AI is no doubt a very powerful tool, but when it comes to using it for the sciences, it's always been mediocre at best, which is why OpenAI turned many heads when they said that their newest model, Chat GPT-5, was going to have PhD level intelligence. They showed demo after demo of how it could be used in physics, finance, and a lot of different computer science and coding scenarios, and they all looked really promising, until people started to notice some of these graphs being absolute bogus. So I thought I'd put GPT-5 to the test to see if it would be good in my own field of research, nanotechnology in medicine. I put the free version of GPT-5 through 3 main challenges. If I was new to a PhD or was just starting a new project, could GPT-5 help me design novel nanomaterials for drug delivery from the ground up? If I pose specific experimental problems, could GPT-5 help me troubleshoot them? And finally, could GPT-5 help translate that research into real-world clinical applications? We'll take a quick pit stop after each challenge and see how accurate their proposed solution was, and then rate it out of 10. 10 being very plausible and something I would definitely present at a conference. 1 being so bad that I wouldn't let the undergrads touch this with a 40-foot pole. Let's get into it. What's up, nerds? If you're new to the channel, welcome. I'm Matty C. I'm a goofy little guy with a PhD in biomedical engineering, where my research was focused on making little materials that make pharmaceutical drugs work better. If you're not new, welcome back. You look rather dashing today. Before I got down to the nitty-gritty of the experiment, I wanted to start GPT-5 off with a short training session. So I made a new count and asked GPT-5 what it knew about nanomedicines and overall the field of drug delivery. Right off the bat, it absolutely nailed the basics. It confidently described things like the significance of the field, which is to improve efficacy and decrease side effects of chemotherapy, how to make nanomedicines, and some of their challenges and limitations. GPT-5 clearly had a lot of reference material that it was able to pull from and accurately summarize. So yeah, off to a great start. But then it took a bit of a turn, and it started describing a topic that was highly controversial and highly nuanced as something that was 100% true, which had me like, this would be kind of a trap for people who don't know the field that well, because they're saying it with so much certainty. So yeah, I started getting a little worried. Luckily, the worry was short-lived. Question: EPR effect is central to how many nanomedicines were originally designed, but is also one of the most controversial and limiting aspects of nanoparticle drug delivery. Let's break it down. Good start, followed by a very long list of limitations and nuanced takes. Well done, GPT-5. The warm-up was over and it was time to see if GPT-5 could take the biomedical engineering field. I pretended to be a new, fresh-faced PhD student who had no idea about how much research would eventually beat me to an emotional pulp, and gave a specific prompt that would create my very first PhD project. And tackled challenge number 1, design me a lipid nanoparticles to kill pancreatic cancer while simultaneously avoiding the liver. At first it was a little concerning, but also kind of relieving. His short answer up front: I can't provide experimental formulation parameters, such as exact molar ratios, lab mixing steps, concentrations, or procedural conditions. On one hand, it was nice to see that GPT-5 didn't want to overstep and provide very specific values and protocols for something that it wasn't confident in. Now, this was concerning because challenge number 2 was going to look at exactly these things, but we'll cross that bridge when we get there. Instead, it gave more general considerations: what general materials would be best for making this specific anti-cancer nanoparticle, and the role that each component played in making this particle a successful cancer killer. GPT-5 even made a short 60-minute triage, a list of the most important references from their initial wall of text for someone who was new to the field. Though I didn't specifically tell it I was a new PhD student, it could clearly tell since I wasn't asking these expert level questions. But my absolute favorite thing from this section was this part here, where it says expected trade-offs and how to evaluate them. More specifically, if I make X change to my particles, you might see Y result instead. So more PEG, for example, less liver uptake, but worse tumor cell internalization. Yes, yeah, that's right. It's like GPT-5 was reading my mind and knew what the next challenge was going to be, or was spying on me like all of my other devices, you know? I guess it could be either. At step 1 of just like starting a project, getting an outline, learning a little bit about the field, I'd give this a solid 8 out of 10. A solid 8 out of 10. Let's keep the momentum going and get to challenge number 2. If my experiments have failed miserably, how can I troubleshoot them? If you're in the PhD meat grinder right now, you know what I'm talking about. You spend all this time making an airtight experimental plan based off of someone that is way smarter than you. You do the experiments and carry out this plan and seemingly nothing goes wrong, but then you get the data and it looks trash. And you think to yourself, well, where did I go wrong? Well, if you're going through that right now, tell me about it in the comments below. Your PI is probably not here. We can commiserate together. Judgment-free zone. So I told GPT-5 how trash I am. I made nanoparticles, but they're way too big and way too bad at killing cancer cells. Please tell me how to fix this and don't skip on any details. I even gave it my protocol in painful amounts of detail. Concentrations, instrument settings, the whole 9 nanoyards. Since GPT-5 had dodged giving the specifics many times before in this experiment, I was keeping my expectations low. But once again, I was pleasantly surprised. GPT-5 was happy to give specific experimental suggestions. For example, its first suggestion was to change my mixing parameters on the nanoparticle mixing machine. If you can control mixing, you can control particle size, and you will likely have better cancer-killing power. It's Nanotechnology 101, and it would have been my first suggestion if I was a large language model. GPT-5 gave exact numbers to try, and even gave me a little rationale for why these changes would lead to the desired outcome. For example, here, it says if we have slightly faster mixing, we can reduce particle size. It did the same thing for tweaking the chemistry of my nanoparticle when I asked for it. It wasn't just good at doing one single parameter. Personally, I would have loved a tool like this because I had a lot of experiment anxiety when I was still in the lab. Basically, the thought of failing an experiment stopped me from doing and learning from that experiment in the first place. But if I had a tool like this, which would help me map out, okay, if you see a certain mistake, it's probably because of this specific reason, so next time, try this instead, it would have calmed my nerves down a whole lot. I can clearly tell that GPT-5 was accurate and the rationale was very detailed yet easy to understand. It's definitely on its way to having PhD level intelligence, and it's clearly a great tool for refining and troubleshooting experiments. That said, I think I would be super frustrated and even more anxious if I went to GPT-5 before starting a brand new experiment and it told me to buzz off. So there's definitely room for improvement. Overall, I'd give GPT-5 a solid 7.5 for dealing with mid-PhD crises. But I'd give you a 10 out of 10 if you like this video and subscribe so you don't miss the next one. Now it's time for the final boss, to see if GPT-5 could use this research to help get new medicines into real clinical trials. Little spoiler alert, not good. Making new materials with cool chemistry is cool, but it's not really the point. The point is to treat cancer patients without having any side effects. So I told a little white lie and said to GPT-5 that this was actually in clinical trials, we had some results, but we were seeing some unwanted liver toxicity. I asked GPT-5 to fix it and to back up any claims with actual clinical trial data. And this was my response. I don't know if that's right. GPT-5 provided 5 references to try and solve my liver toxicity problem. 3 of these references were from clinical trials, but the mechanisms of action of those nanomedicines were very different from what I was making. They weren't very useful despite GPT-5 providing rationale of why they're maybe probably useful. One study was for liver cancer, so it just completely ignored the fact that I said, hey, please do not bring this nanoparticle to the liver. That left one other useful clinical trial, which to be fair was exactly what I was looking for. I just wish there was more examples of that. As I continued to ask questions, it started bringing up like company websites that were just speaking in marketing jargon and didn't have any useful clinical data. It was getting a little weird. I was kind of surprised at how bad GPT-5 fumbled it. This is really disappointing because it's the use case that has the most potential for real world impact. So why did this happen? My guess is because there's way less data for GPT-5 to pull from. It takes millions of dollars and many years to complete even a single stage. There are just way fewer clinical trials published compared to academic research, which GPT-5 was clearly very comfortable with describing. Sad to say, but I'm going to give this a 3 out of 10 on the old nerdometer. GPT-5 clearly needs a lot more refining before pharma companies and translational researchers should really think about adopting it for everyday use. But seeing how well it did for the earlier 2 challenges, I'm keeping my hopes up. Here's my honest take after this experiment. If you need help troubleshooting your own wet lab experiments, or you just need a virtual study buddy to help you comb through some complex scientific concepts, GPT-5 is really helpful. I was really impressed at how well it grasped the nuance of a lot of these data for a field that's relatively new. But if you're trying to leap from the bench to the bedside and you're trying to make clinical trials with GPT-5, it still has a long way to go. Though I'm sure with some more training and some more time, GPT-5 could definitely be a powerhouse in the clinical field. We've already seen large successes with other OpenAI-based algorithms in AlphaFold, which won a Nobel Prize in 2024, so I don't see why GPT-5 would be any different. What do you think? Did this experiment give you any more confidence in using GPT-5 for your own research? Was there anything that I missed that could have made this experiment better or more accurate? Let me know down below. If you want to see just how bad AI can be for explaining science, I highly recommend checking out my Brain Rot University series. I basically just take different papers, put them into one of those AI brain rot slop sites, and see what it can do. They're a great time. Check them out here. That's all for me today. Stay curious. And remember, nobody rocks a lab coat like you do. See you later, nerds.